Thanks for the invitation, of course. And I should have thrown some slides in on C. elegans. I just made the assumption that people would know what it is. It's a microscopic nematode, a millimetre long, and a very powerful system in the aging community. My old girlfriend used to quip that my entire family had the same profile, so I'll just give the cameraman the famous Ray profile. Okay, so let me start out by the key findings of the talk today. I'm going to tell you that the particular class of mutants that I work on, called the mutants, which I'll describe in a minute, excrete a unique set of metabolites, and those metabolites, we think, actually accumulate as a consequence of inhibition of a family of enzymes called the α-ketocid dehydrogenases, and we think that these metabolites are in of themselves responsible, at least in part, for increasing lifespan, and we think they do that via their inhibition of a family of enzymes called the cupins. Okay, so the mitochondrial mutants in C. elegans are a class of long-lived worms. There are different ways that you can get longevity in worms. The ones that I study, that my group studies, affect the electron transport chain and oxidative phosphorylation, either directly or indirectly. Most of those have been defined by RNAi. We're going to be focusing on the genetically defined ones today. The lifespan extension that we see is on the order of somewhere between 20 and 200%. And the mite phenotype is characterized not only by life extension, but by several other phenotypes that can in fact be uncoupled from the longevity phenotype. So this is the kind of life extension that we see in our worms. We're looking at a survival plot here, where we've just got fraction of animals alive versus time, and you can see the control animals at 20 degrees Celsius live on average somewhere between 2 to 3 weeks. But the kind of life extension that we see in these electron transport chain mutants is almost double. And the mutations can be spread all across the electron transport chain. And I'm certainly aware of the fact, although I have the slide at the end. If we get time, I'll go through it. That a lot of mutations in humans that affect the mitochondria result in life shortening, not life lengthening. The answer to that is, in fact, worms are no different. It turns out that you actually need to have mild inhibition rather than full inhibition to get the longevity response. So there seems to be some kind of response going on there metabolically. And this slide is just showing a bunch of different kinds of mutants that are spread all across the chain. Not all electron transport chain mutants in worms are in fact long-lived. The ones that we're going to be looking at today, the three in red, NEO6, ISP1 and CLOC1, are long-lived. The other two that are illustrated here in blue are short-lived. And you can see that ISP1 and UCR2.3 both affect complex 3, and there really is no rhyme and reason for this. So our goal, using a metabolomics approach, was to try and understand why, in fact, longevity results in some of these animals as opposed to shortivity in others. And our hypothesis was very simple, that the mite mutants utilize an altered metabolism, and that metabolism is somehow conducive to life extension. To address that question, or that hypothesis, we undertook a very simple analysis. We knew that in a lot of parasitic species, in the gut, for example, depending on what kind of worm you are, you secrete different kind of metabolites that are indicative, that are a footprint, essentially, of your metabolism that you're running, different kinds of fermentations and stuff like that. So what we did is we cooked up a method to map what we call the excretome of our worms, and we just used basically a GCMS approach. We had a terbutyl derivative, just a basic electron impact ionization. We had to assemble our own library based on, we pieced it together from free libraries, but also we built our own library as well, based on some of the chemicals that we could identify. And so we just did pattern matching. And here's what we have here. So N2 is the wild-type strain, OSP1 is our long-lived strain, and MEV1 is our short-lived strain. And we have, we could detect about 200 metabolites. This has got a mind of its own, it looks like. And we're looking at every metabolite that we could detect. This was untargeted. And we sampled over time. So we sampled all the way from 30 minutes, which was the earliest time point that we could collect, up to 20 hours. And you can see that there are very clearly metabolites that distinguish these long-lived animals from the short-lived ones. The thing that stuck out most to us was that there were a bunch of α-ketoacids and α-hydroxyacids. The α-hydroxyacids are the reduction products of the ketoacids. The enzymes that typically catalyze these reactions are lactate dehydrogenases, and usually they're rate limited by the NAD plus ratio. So their relative ratio reflects, it's in a sense, the redox, not in a sense, they directly reflect the redox potential. Okay, so things that stick out here, as I said, the branch chain ketoacids, pyruvate was in this mix as well, and many of their reduction products. This profile was also seen in OSP1 and CLOC1, another long-lived mutant, and we also saw a distinctive short-lived profile that was also present in our two genetic short-lived mutants. The profile, the long-lived profile, was again distinct from wild-type animals. We also saw this profile in a third mite mutant, long-lived mite mutant, NUO6, and we did not see it in other forms of longevity, other different genetic pathways that are known to mediate lifespan extension in worms, including the DAF2 pathway, the insulin-like pathway. So we have identified these metabolites, so the real question is, do they have any effect, and what's causing them? So we tested several possibilities, but I'm only going to run very quickly through two of those. We first tested whether there was an aberrant activation of anaerobic metabolism in these worms. So C. elegans is unusual in that it can survive 3 days under complete anoxia before it dies. So the simple hypothesis is that these worms have somehow figured out a way to turn on an anaerobic metabolism. The second possibility is the fact that all of these metabolites that we saw building up were related to the enzyme dihydrolyte dehydrogenase, maybe some form of inhibition on that enzyme. So we cooked up our own methods for footprinting worms under normoxia and anoxia. You can see that we've detected a bunch of metabolites using both the derivatization to get them volatile, but also we actually went in and cooked up a second method looking at the volatiles themselves. We didn't even derivatize these things. And it's very clear that with this approach, this was our testbed approach where we could actually distinguish an anoxic metabolism from a normoxic one. So the real question is, do we see this metabolism in our mite mutants? But before going there, just let me say that the mammalian system, we had to pretty much go back and reassemble our own metabolism. There aren't really good maps and databases for the worms. But the take-home message of what we found was that C. elegans is a mixed acid fermenter under anoxic conditions. But most importantly, we found evidence for worms undertaking this unusual reverse flux through the branch chain amino acid pathway. Worms have a rhodoquinone in addition to a ubiquinone in their mitochondrial electron transport chain, so they can actually run their mitochondria in a fermentative manner, utilizing complex 1 and then reversing complex 2, and still taking advantage of a proton gradient to run complex 5. Slightly more efficient than glycolysis, but certainly not as efficient as under oxygen. Nonetheless, the most important thing is, under anoxic conditions, you actually see a reduction in all of the metabolites that we saw accumulating in worms, suggesting that they really aren't running on an anaerobic metabolism. And moreover, you can you can induce the full volatile response when you put mite mutants under anaerobia. So we're pretty certain that they weren't running on an anaerobic metabolism. But here's where the what we think is the most important finding of our work. So dihydrolipid dehydrogenase. If you look at all of the metabolites that accumulated, they're all broken down by three giant complexes called the α-ketoacid dehydrogenases. And those three complexes all have this kind of structure. They're all comprised of about 80 subunits, and they're evolutionarily related. So pyruvate dehydrogenase, branch chain ketoacid dehydrogenase, which breaks down the ketoacid of the three branch chain amino acids, valine, leucine, and isoleucine, and alpha-ketoglutarate dehydrogenase. And you can see that what these guys do, they oxidatively decarboxylate their substrate, the ketoacid, throw off carbon dioxide, attach a CoA, and dump their electrons all the way down to NADH. What's most important is all three enzymes share this common enzyme. These, even though they're all evolutionarily related, it's only this enzyme here that's common amongst all three of them. So a simple prediction is that perhaps this enzyme, DLD-E3, is somehow being specifically inhibited in the long-lived mite mutants. It's kind of important to understand the biochemistry behind how this enzyme works, just so we're on the same page for the next couple of slides. But very briefly, as I said, the substrate is an alpha-ketoacid. My goodness, this is touchy. It's an alpha-ketoacid. It's oxidatively decarboxylated on E1, and that uses a thiamine. That's important, as you'll see shortly. The thiamine transfers the electron pair onto E2, which has a covalently bound lipoamide, and that lipoamide is then dumps its electrons onto NADH, NAD plus to make NADH. So you'll understand that this is essentially a mini electron transport chain. And in fact, this is the biggest enzyme complex in the cell, in the cytoplasm. What's important here is that E3 and E2 are fully reversible, but E1 is not. So if you throw electrons backwards into this complex from NADH, they can get stuck on E2 and become a massive Ross generator. Not only that, and then kill the enzyme. Our first question was, is DLD changed in our long-lived worms? We established an enzymatic assay in the lab to measure DLD activity in our worms. And we synthesized the substrate, and you can see with an RNAi knockdown, the nice thing about C. elegans is that you can feed the worms bacteria that make double-stranded RNA that target your favorite gene. And you can knock down your gene simply by feeding worms bacteria against your favorite gene. So what we've done here is we've just showed that we can knock down our enzyme activity. When we then went in and looked at the metabolic profile of worms fed DLD RNAi, with increasing amount of knockdown, so there's less DLD activity. This is the same as our time profile before. We can see very clearly that our ketoacids accumulate. We actually get a very similar profile to what we see in the mite mutants. So the million-dollar question is, if we now come in and knock down DLD, do we see lifespan extension? Is that in of itself sufficient to recapitulate the mite phenotype? And then we found this quite incredible result. So what we have here is we're looking at DLD RNAi dilution. So actually this is more potent DL knockdown. Rather than seeing a massive lifespan increase, we first saw a decrease and then we saw an increase. And this is probably the only time that I'm ever aware of in the literature, not only in my lab, of seeing a decrease and then an increase in lifespan. So this is kind of very unexpected. Not only that, when we went in to look at and measure DLD activity in our long-lived worms, it really wasn't reduced. So the question is, we've got a profile that's very similar a DLD profile. 3 minutes, okay. It's very similar to our mite mutants, and yet we can't fully recapitulate the phenotype. To cut a long story short, the answer is what I mentioned about the biochemistry. We have evidence that it's the NADH feeding backwards and killing the enzyme complexes that way. So then what we ended up doing was showing that if we used a genetic approach where we knocked out thiamine levels to simultaneously knock down all three of those branched-chain ketoacid, the pyruvate dehydrogenase, and the α-ketoglutarate-dehydrogenase. But what I didn't tell you is DLD has a fourth enzyme that it works in, the glycine cleavage system. If we now come in and just knock out those three enzymes, not only do we phenocopy the profile again, but we also get a very robust lifespan extension. And we did that genetically using this TPK1 mutant to genetically knock out thiamine, and then as a consequence knock down three ketoacids. This just shows when we look at the metabolic profile, there's a very strong correlation between TPK1 and our mite mutants and the DLD neat. Okay, so at the moment what I've told you is that we believe that the buildup of NADH in our mutants, remember all of our electron transport chain mutants are going to be building up NADH simply by virtue of the mutation itself. We believe they're feeding backwards into these alpha-ketoacid dehydrogenases. They're accumulating the alpha-ketoacids, which are then reduced to alpha-hydroxyacids. The million-dollar question is, do these things themselves have a function on longevity? And I'm going to show you the answer is yes. So what I'm showing you here is all of the metabolites that we detected in red and blue that were significantly upregulated in our mite mutants, our long-lived mite mutants. Those in red are also known inhibitors of a family of enzymes called the cupins, and you can see their structural similarity. And also over here we've measured the concentrations, and they're all within the realm of those concentrations that are known to inhibit cupins. What are the cupins? The cupins are a large superfamily of enzymes that all utilize α-ketoglutarate as a co-substrate. You'll recognize many of these names over here. The ones that we're interested in is going to be PHD1, the prolyl-hydroxylase. All of these enzymes work by, they have two obligatory substrates, oxygen and alpha-ketoglutarate, and then they have their own dedicated substrate. Many of the compounds that we detected are known inhibitors of cupins. This is the one that we were interested in. People are most likely familiar with the modulation of HIF1, hypoxia inducible factor. This gene is turned on under hypoxic conditions. Normally it's hydroxylated on a proline and it's degraded by the VHL/E3 ubiquitin ligase complex. Under hypoxic conditions, the lack of oxygen leads to a stabilization of HIF1 and an upregulation of a variety of genes that are required for metabolic adaptation. HIF1 we pulled out in a genetic screen, a transcription factor screen, as being completely required for the longevity response, and you can see that if we knock down HIF1 over here, we don't get the lifespan extension, has no effect on wild-type worms. HIF1's binding partner, AHA1, also come up in our screen. This is an untargeted screen. Here what we're showing is that when we add these metabolites first to worms, we can stimulate the movement of a HIF1 reporter. We can stimulate this, we can stabilize it. When we add these metabolites to a 3D3L1 fibroblast that's got a HIF response element coupled to luciferase, we can stimulate HIF1 activity. Here's what we're showing is when we add these metabolites to another member of the cupin family. This is a epigenetic modifying enzyme, JmjD2A. We can also inhibit it. We found, I'm not going to show you the data, that these metabolites are broken down by bacteria. We have to grow the worms on bacteria to do lifespan assays. It took us a long time to figure it out. The literature is replete with a lot of, unfortunately,
probably data that's not correct. We found that 2,4-PDA was a known inhibitor of the cupins, and it was stable under our assay conditions. When we added it to worms, we can increase their lifespan. It's dose dependent, it's HIF1 dependent, and this is my last slide. Here's what we think's going on with the longevity in these worms. We think that, based on our studies, when the NADH builds up in our mutants that have electron transport chain defects, we feed electrons back into the alpha-ketoacid dehydrogenases. The alpha-ketoacids accumulate, and then those ketoacids then go on to affect the cupin network. And again, just summarising the key findings and thank people.